Hello everyone, welcome to the channel Microbial World. Today's topic is Helicobacter pylori. Morphology and habitat. Helicobacter pylori is a spiral-shaped or helical bacterium. It is a gram-negative bacilli, which means it has a thin peptidoglycan layer and an outer membrane containing lipopolysaccharide. It is motile with Lophotrichus flagella. Lophotrichus flagella is a flagellar arrangement where multiple flagella is present at one pole or end of the bacterium. Helicobacter pylori lives in the mucosal lining of duodenum and stomach. It is the spiral shape of the bacteria that helps it to penetrate the stomach's mucosal lining. And the Lophotrichus flagella helps the bacterium to move in the viscous environment of the stomach's mucus and aid in its colonization. Cultural characteristics. Helicobacter pylori is microaerophilic, which means it requires a low oxygen environment to grow. It can grow in environment with reduced oxygen levels around 5% oxygen, 10% carbon dioxide and 85% nitrogen. It grows optimally at a temperature of 37 degrees Celsius and an acidic pH range of 5.5 to 7. Helicobacter pylori grows best on enriched media like chocolate agar or blood agar, which provides essential growth factors. Colonies are generally small and it takes 2 to 7 days to develop. Pathogenesis. Helicobacter pylori usually enters the stomach through contaminated food and water and colonizes the gastric mucosa. It has many adaptations that helps it to survive the harsh acidic environment of the stomach. One of the adaptations is the production of urease, an enzyme that breaks down urea into ammonia, neutralizing the surrounding stomach acid. After neutralizing the stomach acid, Helicobacter pylori attaches to the epithelial cells of the stomach lining using specialized adhesion molecules. This adhesion helps the bacteria to avoid being flushed out by the stomach's mucus or peristaltic movements. Helicobacter pylori produces virulence factors. CAG-A, cytotoxin-associated gene A. CAG-A is injected into the gastric cells. It disrupts the normal cell function and can lead to inflammation and an increased risk of gastric cancer. VAC-A, vacuolating cytotoxin A. This toxin induces vacuole formation in the cells and can cause apoptosis, disrupting the integrity of the stomach lining. The immune system responds to the infection by sending immune cells to the stomach lining. But Helicobacter pylori has mechanisms to evade the immune response and can lead to chronic inflammation, gastritis. Persistent infection and inflammation can damage the stomach lining over time. This combined with stomach acid can result in ulcers in the duodenum or the stomach itself. Symptoms of Helicobacter pylori infection includes abdominal pain. This is commonly felt in the upper abdomen region and the pain worsens when the stomach is empty. Bloating. A sensation of fullness or swelling in the abdomen, often after meals. Infection also causes a persistent feeling of nausea that leads to vomiting. Another symptom is loss of appetite. Excessive belching or burping. Frequent burping occurs due to excessive gas production in the stomach. Heartburn is another symptom. This occurs due to acid backing up in the esophagus and causing a discomfort in the chest. Dark colored stools indicates gastrointestinal bleeding. Fatigue is a less common symptom and it occurs due to anemia from blood loss. Lab diagnosis. Non-invasive test is often the first choice for diagnosing Helicobacter pylori infection as it does not require an endoscopy. Urea breath test. In this test, the patient ingests urea labeled with a radioactive or stable isotope. If Helicobacter pylori is present, it breaks down the urea to release carbon dioxide labeled with the isotope. The labeled carbon dioxide is detected in the breath. Stool antigen test detects Helicobacter pylori antigens in the stool samples. Serology involves antibody test to detect IgG antibodies against Helicobacter pylori in the blood. Invasive test often requires an endoscopic examination and it is used when upper gastrointestinal endoscopy is clinically indicated, for example in case of ulcers or suspected cancer. Rapid urease test. In this test, a biopsy sample from the stomach is placed on a medium containing urea and a pH indicator. If Helicobacter pylori is present, its urease enzyme breaks down urea into ammonia, changing the color and pH of the medium. This is a very quick and reliable test. In histology, biopsy samples are examined under microscope using special staining methods such as Giemsa staining to identify Helicobacter pylori. Polymerase chain reaction PCR detects Helicobacter pylori DNA in gastric biopsies or other samples. The treatment of Helicobacter pylori infection involves a combination of antibiotics and acid suppressing medications. The standard treatment includes the usage of bismuth salicylate, tetracycline or amoxicillin, and metronidazole. The first-line treatment is the most widely used treatment method and it involves the usage of antibiotics such as clarithromycin, amoxicillin, and metronidazole. The success rate for this method is 75 to 80%. If the first-line treatment fails, second-line treatment is carried out, which involves using bismuth salicylate, tetracycline or nitroimidazole. The success rate of second-line treatment is 85 to 90%. So this is all about today's topic. I hope that the video was useful for everyone. Thank you for watching.